Okay, let's start talking about Chapter 14. The first topic here is going to be talking about karyotypes. So what is a karyotype? A karyotype shows the complete diploid set of chromosomes grouped together in pairs, arranged in order of decreasing size. Remember that diploid also means 2N. So we're looking at both sets of chromosomes. To find what makes us uniquely human, we have to explore the human genome. A genome is the full set of genetic information that an organism carries in its DNA. A study of any genome starts with chromosomes, the bundles of DNA and protein found in the nuclei of eukaryotic cells. To see human chromosomes clearly, cell biologists photograph cells in mitosis when the chromosomes are fully condensed and easy to view. Scientists then cut out the chromosomes from the photographs and arrange them in a picture known as a karyotype. It shows the complete diploid set of chromosomes grouped together in pairs, arranged in order of decreasing size. A karyotype from a typical human cell, which contains 46 chromosomes, is arranged in 23. 2 of the 46 chromosomes in the human genome are known as sex chromosomes because they determine an individual's sex. Females have 2 copies of the X chromosome. Males have 1 X chromosome and 1 Y chromosome. This Punnett square illustrates why males and females are born in a roughly 50-50 ratio. All human eggs carry a single X chromosome. So there are 23 chromosomes, and that 23rd one is going to be an X. However, half of all sperm cells carry an X chromosome, and half carry a Y chromosome. Okay, so 23 in the sperm, but the 23rd is a Y. This ensures that just about half the zygotes will be males and half will be females. Since females carry XX and males are XY, when we do Punnett squares, okay, we see that half of the children can be female and half of the children will be male, which is typical. We know that when a woman is pregnant, there's a 50-50 chance that she'll have a boy and a girl. More than 1,200 genes are found in the X chromosome, some of which are shown here. The Y chromosome is much smaller than the X chromosome and contains only about 140 genes, most of which are associated with male sex determination and sperm development. So we see how much smaller the Y chromosome is. The remaining 44 chromosomes are known as autosomal chromosomes or autosomes. The complete human genome consists of 46 chromosomes, including 44 autosomes and 2 sex chromosomes. To quickly summarize the total number of chromosomes present in a human cell, biologists write 46 XX for females and 46 XY for males. So right here, those are going to be the sex chromosomes. Okay, so I'm going to abbreviate, so sex chromosomes. Everything else, okay, all those, that's going to be the autosomes. Every single cell, so all the cells that are diploid and have 46 total chromosomes are going to have sex chromosomes as the final pair. Again, here this is showing XY, so that's going to be a guy. If it was a female, we'd see 2 of the same size. There'd be 2 X's. But everything else are going to be called autosomes. So notice in this karyotype, again, we're having everything is lined up and paired up from biggest to smallest, and then we put the sex chromosomes at the end. There are a couple of things I want to point out here just to sort of connect this karyotype picture to what we have learned in the past. So let's zoom in here, okay? 1, these are homologous chromosomes, that word that we love, right? So this one maybe came from mom and this one came from dad, but they're both chromosome 1, they're coding for the same thing. And remember, chromosomes are made up of 2 sister chromatids, right, held together in the middle. So this right here is a chromatid, and this right there is another chromatid, right, held together in the middle by a centromere. So I think these karyotypes really help us to see, yeah, this is a homologous chromosome from mom, from dad. And then in meiosis, these guys, your homologous chromosomes, are going to line up in the middle and they're going to separate, right? So when they're in the cell, you're only going to pass one on to your child, okay? And which one it is, that's why we do Punnett squares, to see, is it this one that has like the big B, or is it this over here that just has the little b? And then that's, so hopefully that just connects from what we've learned to this picture right here. The other thing that's good for karyotypes is that, one, it's going to be telling doctors and genetic counselors the sex. So again, so they'll be able to look and say, oh, this has XY, so that's going to be a boy, and if it's XX, then they know it's a girl. So karyotypes are good for that. Karyotypes are also good for folks looking at it and making sure that there's the correct number, and if there's not the correct number, then that's where they can start to figure out which genetic disorder somebody might have. So do they have an extra number 21? Is there like an extra one? Or are they missing a chromosome? Are they missing part of a chromosome? Like maybe like a section of a chromosome is just deleted and it's not there at all. So karyotypes are good for all of that as well. All right. So I hope you've learned a bit about karyotypes. If you have any questions, feel free to ask. Make good choices. Have a good day, and Jesus loves you.